Good morning, everyone. Welcome to the parallel session in chemistry and biochemistry. This event is organized by the Philippine-American Academy of Science and Engineering. We are celebrating the 40th PAASE anniversary and 2020 annual scientific meeting with the theme, Outreach to Philippine Regions. I am Gladys Completo from the Institute of Chemistry, UP Los Baños, and I am your host for this session. With me as co-hosts are Dr. Ruel Nacario of UP Los Baños, Dr. Isagani Padulina of Pascual Pharma, and Dr. Jeffrey Buncuin of Exxon. I am greatly honored and privileged to host this webinar. We have an excellent lineup of speakers, ranging from the rising stars of the field to established, respected, multi-awarded scientists and trailblazers in their areas of expertise. So we have two special guest speakers from Academia Sinica, Dr. Todd Lowary and Dr. Hans Lin. And for the actual parallel session, we have two successful Filipino scientists that are based in the US, Dr. Roy Herona and Dr. Rodel Barrientos. And last but not the least, we have Dr. Michelle Rigolacio from UP Diliman. So before we start the webinar, let me present some reminders for the attendees. Okay, so these are the reminders. Please make sure that your gadget is on mute. And then if you joined via Zoom, please write your questions in the Q&A box. And then questions will be presented by the co-host during the open forum. And if you are watching the YouTube live stream, please write your questions in the live chat. And again, these questions will be presented during the open forum. And if you want to ask questions live, you can click the raise hand button and wait for the moderator to entertain you. So let me start by introducing the first speaker. Okay, Dr. Todd Lowary. Dr. Todd Lowary is a professor at the University of Alberta and currently a distinguished research fellow and the director of the Institute of Biological Chemistry at Academia Sinica. His research interests are in the synthesis of novel probe molecules for understanding the role of carbohydrates in biology, in particular infectious diseases. Dr. Lowary garnered several prestigious awards for his work, including the Raymond Lemieux Professor of Carbohydrate Chemistry, Tier 1 Canada Research in the Department of Chemistry, Alfred Baylor Award for Organic Chemistry, and this year he was awarded the Arthur C. Cope Scholar Award from the American Chemical Society, given out each year to recognize excellence in organic chemistry. He was recognized for his contributions to the development of novel methods for the synthesis of microbial carbohydrates and application of those methods to complex glycan synthesis and biology. Todd Lowary received his bachelor's degree in chemistry from the University of Montana and obtained his PhD in organic chemistry under the supervision of Professor Hinsgall at the University of Alberta. Let us all welcome Dr. Todd Lowary. You're on mute, Todd.
Okay, so you can see my screen and hear me? Okay, yes. So you can see my screen and you can hear me. Is that right? Is that right? I can't.
Yes.
Yeah, we can hear you and see the screen.
All right, sorry, this is the first time I've done this in a while. So thank you very much, Gladys, for the invitation to speak in this event, and also thank you for the very nice introduction. So today I want to tell you a story about a project that's been going on in our lab for a long time, which is dedicated to trying to understand how the host responds to infection by mycobacteria. And the most famous Mycobacteria is Mycobacterium tuberculosis, which, as all of you are probably aware, is an important bacterial disease, which is particularly a problem in the developing world. And so today what I'll tell you about is work that we've done trying to understand this immune response, and then also how that's actually leading to some, I think, potentially impactful work in the area of diagnostics. So this is a cartoon structure of the mycobacterial cell wall. It's a quite complex array of structures. It has a layer of peptidoglycan, a large structure called the Mycobacterial complex. And then the outer periphery of the cell wall then are a number of carbohydrate structures. And this will be the focus of the talk today is the carbohydrate structures that are shown out here. So these are the sugars and the species that actually are the first thing that your immune system interacts with when you're infected. And so not surprisingly then, these are important players in the way that your immune system reacts to these bacteria. So in the next couple of slides, I want to give you a sort of a flavor for what the structure of these molecules are so that you can understand sort of the chemical diversity of the structures. And frankly, the reason that we got interested in this problem initially was really because we found the structures quite intriguing. So the major structures in our family of molecules called extractable glycolipids, and then another molecule called Liporabinomanon or LAM, or a delipidated version of that called arabinomanon. So the extractable glycolipids then, examples of those are shown here. There are three major classes. There's one family called glycopeptidolipids, which, as the name implies, it involves are molecules that have sugars, peptides, and then lipids. There's another group called phenolic glycolipids, which have then a glycan attached then through a phenolic group to a very complex lipid. And finally, the last group is what's referred to as lipooligosaccharides, which consist of a trihalose core attached to some azo groups, which is then attached to a species-specific glycan. These have very unusual structures, generally speaking. And so, for example, this is kind of an interesting monosaccharide where you have a branch chain sugar where off of one end of the carbohydrate, you have then a polyhydroxylated chain. So one of the things that struck us about these were the unusual nature of the structures of the sugars, many methylated sugars, as well as I pointed out, some unusual structures. And these are found in a range of different organisms, and so some of them are found more commonly in others than in, for example, in TB. But nevertheless, they are present widely across the different mycobacterial domains. Something common to all mycobacteria is this structure called liporabinomanon or arabinomanon, and this is a cartoon structure here of this molecule. Each of the shapes refers to a carbohydrate residue. So this is attached at the one end to an acyl group, which has a diacylglycerol moiety, and in the arabinomanon, this lipid chain is then missing. This consists then of a chain of mannose residues that are predominantly linked alpha-1,6, and this is then attached to a chain of arabinofuranose residues, which are generally linked linearly with occasional branch points. The ends of this are then capped or terminated with two different motifs. These are referred to as either the ARA6 motif or the ARA4 motif. And so these are structurally related, as you can tell, with the ARA4 motif just being sort of a fragment of this complete structure here. The terminal hydroxyl groups then on these structures then serve as the attachment site for different capping motifs. So here's an example of one of the capping motifs. It's something we refer to as the MTX motif. These capping motifs then are specific to different species, and the examples that have been identified to date include the following. So in many pathogenic species, for example, TB, bovis, which is a cattle disease, avium and lepre, these are short mannose oligosaccharides. Sometimes these mannose oligosaccharides are functionalized with this MTX motif, and this is going to be an important part of the discussion later today. They can be capped with inositophosphates, or in some cases, there's no capping at all. And so, again, we were intrigued by the structures of this, but perhaps what's more interesting and important really is how these molecules interact with the immune system. And so it's known that these glycans then are ligands for lectins of the innate immune system. It's also known that they're very potent inducers of antibodies. But generally speaking, there's a very poor understanding at the molecular level of how these glycans mediate their biological effects. And so, for example, although there are a number of anti-LAM monoclonal antibodies, until the work that I'm going to tell you about today, the specificity of only one was known. And this is an antibody called CS35. Several years ago, we published a crystal structure of this antibody bound to its ligand, which is this hexasaccharide. And so this then provides us with some detailed understanding of how these types of glycans are recognized by by the immune system. But we wanted to better understand this and to probe this in greater detail across a wide range of structures and across a larger range of antibodies. And so the idea was to identify a series of targets, make these targets using synthetic chemistry, generate the molecules, and then print them on arrays, and then do ELISA type structure studies to do structure function studies. So we set off to make initially a target of 75 glycans. These include different fragments of LAM, the extractable glycolipids, and then a couple of structures I didn't mention to you. We're not quite at 75 yet. We're actually at 62, although we've made a few more that we haven't incorporated onto the array. So if you would think about perhaps how you would go about trying to access these glycans, you could think about two different approaches. One would involve the synthetic approaches, which is the one we've used. So the advantages of this are that you can actually produce well-defined molecules that you know the structures of. You can often isolate these in higher overall yield than those you can get out of nature. It's easy to make fragments of structures for probing structure-activity relationships. We can make milligram or even in some cases gram amounts of materials, which is, I would say, more or less impossible through isolation. And importantly, we can actually generate molecules that are tagged with groups that allow their conjugation to other materials. So for example, a protein or to a surface. So those are some advantages of a synthetic approach over, say, isolation of these compounds from nature. Of course, the disadvantages of a synthetic approach are that it takes a lot of work and it requires a level of expertise, but that's what we do is we make glycans. And you also have to know what it is that you want to synthesize. You simply can't start making compounds. And so what we do is we rely on the literature to tell us what types of structures are in these glycans, and that's then determines what we try to make. So there's a lot of interest currently in using enzymes to make glycans, and in some areas of carbohydrate chemistry and biochemistry, this is extremely powerful and very well developed. Unfortunately, mycobacterial glycan enzymology is not very well understood yet, and so at this point, making these types of structures using enzymes is really not an option. So I won't talk about any chemistry today. I just want to talk about what we've done with the molecules. And so this is the array that we did the work that I'll tell you about today. This was a smaller version of the array. It had only 60 structures. So there are a series of LAM fragments, a series of phenolic glycolipids, and then a smaller number of the trailless mycolates and lipoligosaccharides, these glucans that I didn't really mention, and finally a small number of these glycopeptidolipids. So we've got in total 60 compounds. With those compounds in hand, we move forward to try to screen then a panel of monoclonal antibodies that recognize LAM. And so the discussion today will focus solely on LAM. So again, LAM is an important antigen. If you are infected with Mycobacteria, you'll have very high titers of anti-LAM antibodies. And a monoclonal antibody that's used, that recognizes LAM has actually been used in a diagnostic method for TB. And basically this works by detecting the presence of LAM in urine from patients. And I'll come back and talk about that a little bit later in the talk. As I mentioned earlier, the specificity of only one anti-LAM antibody before we started this work was known, and that was CS35. And so we did is we screened in collaboration with people at Rutgers University and the Foundation for Innovative New Diagnostics, a panel of antibodies. These include many from mice, others from human, and some from rabbits and chickens. I should point out that the human antibodies are actually isolated from B cells of TB patients. And so these are ones that have some potentially clinical relevance. So with the array then, what we can do is we can screen then the affinity of these different structures for the compounds. And what I show here is a plot of the different compounds along the x-axis versus simply the intensity of the signal that we get upon binding. So the higher the signal, then the more intense, the more strongly the binding of the glycan or the protein to the glycan. We look at a range of different proteins here, and this just is done so that we can get a relative sense of affinity. And so what you see here for each antigen is in a mixture of 3 different concentrations at 10 micrograms per mil, 1 microgram per mil, and 0.1 microgram per mil. So what drops out of this, just looking at this data, is that some are very broadly reactive, that react to a large number of things, and others are very specific in terms of what they recognize. And so to give you some idea of what type of structures are important here, these structures here are these types of molecules where you have then all only arabinofuranose residues with occasional branch points, but largely linear structures. The structures here then are more complicated. They're all branched. They contain these sort of hexasaccharide type of motifs, and some of these are quite large structures as well. And these also have these capping motifs that are recognized. And finally, these structures here then are all linear molecules that aren't branched, that contain then these capping motifs, either the mannose or the mannose with the methylthioxylase residue. So if you look at all these structures and say, is there a common structural element? The answer is yes, and that element is this structure shown here. This disaccharide motif is present in all of these. This is this type of a motif where you have two arabinofuranose residues linked then in a beta linkage to the 2 position. And this group can either be an OH group or an R group. That's quite critical. And so, for example, if you look at structures that are very similar to this large glycan here, for example, these 2 compounds. So these are fragments of this where what we've done is simply remove these key motifs. These bind extremely weakly to these antibodies. And so, for example, here is where those bind. So basically you see no binding, only a tiny amount of binding in the case of this one antibody. So even with very large structures, it's clear that you need this one disaccharide motif to be recognized by these antibodies. Here's again some additional data. I want to highlight just a couple of things here. So again, this highlights that many of these are very broadly reactive, but some are quite specific. So 2 that I particularly like, or actually say the one I particularly like, or the most like, is this one. This one binds only to molecules that contain this 5-thiomethylxylose residue attached to the mannose, and so it doesn't bind anything. If it doesn't have this group, it doesn't bind. So this is a specific antibody for this type of motif. Similarly, this type of structure here binds only to these 4 compounds, which then all share this common motif here. So it's very clear from this data what these bind to, but on the other hand, for some of these compounds, it's not clear. Some of these antibodies, it's not clear. And so, for example, this one binds to structures in the core, but it also binds to structures in the termini. And so this is an example where we'll need additional information from structural biology to try to understand how the interactions of, what type of interactions are between these glycans and these proteins. Okay, so I want to wrap up by talking about some application of this, and this is in the area of diagnostics. And so I would guess the current pandemic crisis has underscored the need to have effective diagnostics for any disease, and particularly for those that are highly contagious. So the worldwide diagnostic market for TB is huge. It's more than $1 billion annually, but there really is no perfect test currently for TB. And so the existing diagnostics are either expensive, or they're time consuming, or they're not readily carried out in developing areas, which for a disease like TB, which is largely a 3rd world problem or developing world problem, is an important challenge. And remarkably, many of these are also terribly unreliable. And so there are lots of TB tests on the market that actually are very poor tests. The gold standard, what is used widely, is what's known as the gene expert test. And this employs sputum as the fluid from which the diagnosis is made. This costs about $10 a test, which doesn't sound like a lot of money in North America, but it is a lot of money in many parts of the world. And the only reason it's that inexpensive is that every test is subsidized by the Gates Foundation. And so this is based on nucleic acid amplification, so similar to the COVID type tests that are currently used for detecting the presence of the viral DNA. So this works, but it's not an ideal test. And one of the other challenges about it is that sputum is not an ideal fluid for diagnosing TB, particularly in children. And if you have extrapulmonary TB, it can't be used at all. And so there's a real need to be able to do this with other bodily fluids. So the most easily accessible bodily fluid is likely urine. And so there is actually a urine-based TB diagnostic. And this is called the allele-determined TB test. And this does work. The challenge is that it's only effective in HIV-positive individuals. The reasons for that aren't exactly clear, but it's believed that it's only in people that have suppressed immune systems, where you have large enough amount of bacterial growth that you get sufficient antigen shed into the urine that it can be detected. And so basically you need a fairly high concentration of LAM in the urine, and that appears to be present only when you have people who are immunocompromised, like HIV-positive individuals. So this begs the question then, is if we were able to identify more sensitive monoclonal antibodies, or perhaps a pair of monoclonals that could be used in a CAPS or ELISA type test, would we be more effective, and could we develop a more sensitive assay? And the answer to that question is yes. And so we've been involved in a study that's led to a number of papers over the last 3 to 4 years. on this problem. One of these was just accepted fairly recently. And the idea really is that we were trying to develop a sandwich ELISA type assay. And so our contribution to this has been the work on characterizing these monoclonals. The actual diagnostic work is being done in collaboration with the Foundation for Innovative New Diagnostics, which is a Gates Foundation affiliated NGO based in Geneva. And so the idea is actually a very simple test. And so what you do is you immobilize a capture antibody on a strip. You then treat that strip with urine. The urine has lamb in it. The lamb then binds to the antibody. You then go in with a detection antibody, and then upon that binding event, you can measure this by electrochemical luminescence detection. So this is an assay being developed in collaboration with both FIND as well as the film company in Japan, Fujifilm. So I didn't realize this until fairly recently, but Fujifilm has gotten into the diagnostic business, and so they're a big player in this field. And so basically what we did was we had these antibodies that we screened. We understand the specificity. And then FIND took 100 different pairs of these different combinations of these capture and detecting antibodies to try to identify which were the best pair of antibodies. So what came out of this was that the best capture antibody was this structure here, where this antibody which recognizes all of these beta-RF, 1,2-RF containing glycans. And then the best detection antibody was this structure, which is the one that's MTX specific. And so this is the form of the assay that we've been using. I want to share with you a little bit of data on the success of this assay. And so when we talk about a diagnostic, we talk about two terms. One is sensitivity, and the other is specificity. And so a diagnostic that has high sensitivity, that means it has a low false negative rate. If it is high specificity, that means it has a low false positive rate. And so ideally, you want a diagnostic that has both high sensitivity and high specificity, so you don't have any false positives or negatives. So what I do is compare in this table then the ALIR-TB work, which is this is now the commercially available assay that's used, and then the Fujifilm assay, which is the one that we've been involved in developing. So first talk about HIV positive people. What you can see is that the sensitivity of our test, the Fujifilm test, is actually substantially higher than in the ALIR-TB test, and the specificity is perhaps a bit lower, but fairly comparable. So that's I think interesting. What to me is much more exciting is that when you look now at HIV negative individuals, this test performs much better. And so in the case of the ALIR test, you get only 13% sensitivity. The Fujifilm test, we go up to 80%, and the specificity remains the same. So that I think is a very exciting sort of step forward in this. And of course, then it leads us to the next question: is that can we do even better? And so what we're doing now is we're remaking or making new glycans that contain that MTX residues, which seems to be critical for this. And then those are being used to generate new anti-LAM monoclonal antibodies. So we have the first set of data on this, which unfortunately I'm not in a position to share with you, but it looks like we've identified a couple of new antibodies that have much higher specificity for particular glycans. And now we're in the process of working with FIND to incorporate those antibodies into these diagnostics to see if we can further increase this. So this has been a fun project to work on. And I guess from the student perspective, what I think is nice about this project is that frankly, we started this whole project because we thought these were cool structures. And we were interested in the science, just from a basic science perspective. And what we've been able to do is actually working with the right partners, convert this into a project that involves a number of, I think, interesting clinically relevant aspects that I think makes this potentially a really super impactful sort of study. So with that, I'm going to finish up and just thank the key people in the group. I'm in the process of relocating to Academia Sinica in Taiwan. I still have a small lab in Canada. This was a picture of the lab taken a few years ago in Canada. We would go to the mountains every year for a meeting. The key people involved in this are these individuals shown here. Doing the synthetic work here is a big task, and that was done over many years. And so many of the people that were involved in that synthetic work were not present in the group picture at this time. And then finally, just to thank the people who we work with on this. I mentioned FIND a couple of times, as well as Rutgers. We have other collaborators on other aspects of this project, and we also work with Delphi Chatterjee at Colorado State. A big funder of this has been the Gates Foundation, find, and then other money has come from the Canadian Glycomics Network and CIHR, which is the Canadian equivalent of the National Institutes of Health. With that, I'll stop and I'll be happy to take any questions.
Thank you very much, Todd, for that comprehensive and insightful presentation. Now I give the floor to my co-hosts, Dr. Ruel Nacario and Dr. Gani Padulina, for the Q&A portion.
Hello, Todd. Thanks for that interesting talk. So one of the questions here is that, is it possible to use your technique to distinguish between different mycobacterial species like MTV and, let's say, M. bovis or M. leprae?
In principle, yes. There are structural differences in the glycans between the different mycobacteria. I would say that LAM is not a good choice for that because the LAM molecules are present in all the mycobacteria, and so but what is possible is to use some of the other antigens. And so, for example, I mean, it's not true for bovis, but there's a diagnostic that's for Mycobacterium leprae or leprosy, which is based on an antibody detection, the presence of an antibody that recognizes a specific glycan. So yes, I think there is potential for using these glycans as diagnostic markers of different diseases, but LAM would not be the antigen I would go with. I would go with some of the other structures that are more specific to the different species.
Hello, Todd. We've got a question from the audience. You have about 311, more than 300 participants joining us right now. I'm going to allow Sheila May Bulayutan to talk, okay? Sheila? Go ahead. I think you're on mute, Sheila. Go ahead, Sheila. Okay, while she takes care of the technical stuff, I'm going to ask Maridel, also Maridel Oblad also wants to ask a question. Maridel, I'm going to allow you to talk. Go ahead. Please unmute before you talk. Is UPLB ITC here? Maybe we can help them unmute. They're still on mute, both of them.
I'm actually unmuting them now. They're not responding.
Oh, okay.
Well, I think it's remarkable that there's 300 people up at 8 a.m. to listen to me talk. That's
like
incredible.
300, almost 320 right now.
Yeah. Okay.
Yeah.
And we've got people over YouTube as well, so there's a lot more.
Why don't you go ahead, Jeff? While we
so
wait for these two.
we don't have questions from YouTube as of yet.
Okay.
Yeah.
So I have a question. Dad, so did you try changing that tile group with some isosteres, and then is it still being recognized?
No, we haven't done that. I mean, that's a possibility. One of the things I skipped over is that sulfur can actually be oxidized in situ to the corresponding sulfoxide. And an open question is whether those antibodies actually recognize the sulfide or whether it also recognizes the sulfoxide form. And so that's one of the questions that we want to do with these antibodies is to try to understand what is the nature of those interactions. But possibly another isostere would be interesting. Although from an antigen detection perspective, it's not so relevant because that's not produced naturally. But it would be interesting to study the nature of the interaction.
Yes. There's an assay, I think, that you can use so that you will know if it gets oxidized. And then you can check first.
Yeah, you can see it by NMR, actually. If you do NMR, if we store these over time, slowly you start to see new peaks showing up, and then you do a mass spec, and then it shows that it oxidizes. So, yeah. Good.
Yeah. Okay. Ruel, are there other questions?
There's a question here in the Q&A box. I'm not sure if this is, so I'm reading it here. What may tend to happen if lamb may use in dialysis or other health-related issues?
So the question is if
you
you're
can
on
use,
dialysis?
yeah, if you're in dialysis.
Yeah, that's
If
a
you
good
can
question.
use that.
Yeah, that's a good question. I don't know. Yeah, I don't know. So I'm presumably if you were on dialysis, you would filter out some of the lamb from your urine, and maybe that would make it less sensitive. But that's I don't know the answer to that. And as far as I know, no one's ever, certainly with regard to this assay, no one's compared, say, the sensitivity of the test to people on dialysis versus who aren't. But that's an excellent question, actually. Yeah, I don't have an answer.
I thought I have a question. By the way, thank you for your talk, and it's very interesting. So I have a question about the glycan array.
Yeah.
So maybe I missed it or something, but how do the glycans attach to the surface of the glycan array? So I have two questions. That's the first question. And then the second, yeah. And then the second question is, are those glycan arrays commercially available that other researchers could use probably?
Yeah. So the way we do the conjugation is when we synthesize the glycans, they all have an amino group at one end, and so then we use standard amine type of chemistry to couple them to the array. So there's nothing fancy or special about that. Very, very standard approach. They're not commercially available, but certainly we're open to collaboration. And so if people are interested in accessing it, we would be delighted to work with people. And so, yes. So, but at the moment they're not commercially available. And to be frank, I don't think at this point there's probably not from a research perspective, probably not a huge commercial demand. And so I doubt they'll ever be commercially available. But certainly we're open to collaboration if people are interested.
All right, thank you so much.
Mm-hmm.
Hey, Todd, there's another question from the Zoom Q&A. How long would it take for a similar sophisticated assay to be developed for COVID-19 antigens, given that the antigenic portions are not yet well known?
Yeah, well, I mean, the two diagnostics for COVID at the moment, the one that's used for detecting whether you're currently infected is of course a nucleic acid based test, which is very different than what we're talking about.
Yeah.
There's also the test that measures whether you've been infected or not in the past, which is an antibody test. I don't see that there's a lot of potential for the kind of approach we're looking at here in terms of COVID, in terms of detection of. I don't think any of the current tests that are out there actually detect the spike protein directly. They detect the presence of the antibodies to the spike protein, or they detect the presence of the viral nucleic acid. And the problem with developing an assay directly to the spike protein to the other proteins is as you point out, most of these things are glycosylated, and there's a pretty heterogeneous level of glycosylation, which would complicate the binding assays. Certainly there is an understanding of what glycans are found now on the spike protein, but again, it's quite heterogeneous, and I'm not sure this would be such a good approach for COVID.
Okay, so is it just how hard is it in general to kind of find that? So you're looking at the patterns, right, and seeing what's the probably the most common denominator or something.
Yeah.
So how I guess I'll ask the questions for a lot of the students that are with us right now. What kind of skills do you need to help with that?
I mean, the people that have done all the work are they're trained not as chemists but as microbiologists or biochemists, I would guess, and or lab clinical lab degrees, and so you need skills in doing analytical tests and things of that sort, and then also statistical analysis of the data. And so I would say for the front end of this, making the array, you need to be a chemist. For the back end, you don't need to be a chemist at all, although it's helpful because if you can understand the structures, you can start to learn something more. But to actually look at the data and try to determine what pair of antibodies is the best, that's really just purely a sort of a lab testing, statistical analysis kind of data.
Okay. Thanks, Todd.
Yeah.
Hey, Todd. Roy here. I have a question. First of all, thanks for the very informative talk. I'm not very familiar with the microbiology of multi-drug resistant TB, but I was wondering as I was listening to your talk, is there a possibility to harness the technology to use it for early detection of potential MDR TB cases?
Possibly if there were glycan structure differences in the glycan structures, and I don't think that's never, to my knowledge, being looked at. And so I think most of the studies where people are looking at MDR are based on these nucleic acid amplification tests, where you can look at the emergence of markers that indicate that there's drug resistance. And so my guess is that this approach is not going to be ideal for looking at drug resistance unless there's a dramatic change in the glycan structures, and there's no evidence to suggest that's the case yet.
So that is not known yet?
No, it's not known, and as far as I know, it's not ever been looked at. There's doing the structural work around these is quite challenging, and so it's not easy to get at the structural data. And so that's really a kind of a low that's kind of a barrier to this. Whereas, sequencing DNA can be automated, and so I'm not saying there's not a possibility. It's just that I think it's probably a long way down the road if it were to happen.
Thank you.
Another question, Todd. So in COVID-19, they have this, what they term as the glycan shield, so shielding the spike protein. So do you have a similar? Is there a similar thing in MTB?
Well, I mean, in effect, the cell wall is a glycan shield that hides the organism from the host, right? So you could think about it in a sense, the whole cell wall structure being kind of a glycan shield. I mean, the big difference between the COVID virus and TB is that COVID produces these glycans by taking the biosynthetic machinery of the host, the human, and making its glycans, right? The COVID virus doesn't have its own carbohydrate processing enzyme. So what that means is that the glycans that COVID produces look like human structures, whereas TB have its own system to make its own glycans, and so they're very different, right?
Thank you.
Hey, Jeff, anything from YouTube?
We don't have anything from YouTube yet.
Okay.
Okay, so is that it? No more questions?
Good. Well, thank you all for organizing this, and I appreciate the effort that you all put into this. And salamat po.
Thank you. Salamat, Todd. Thank you very much. We know that you're very busy, and we're very fortunate that you were able to find the time to give this lecture.
My pleasure. All right.
Okay.
Thank you again. Bye-bye.
Yes.
Thanks, Todd.
Thank you, Todd.